Hello everyone, welcome back to another episode on genetics. Today we're going to do some gene linkage practice problems, and there should be sort of a step-by-step way how you can solve some of these problems that you might see like on an exam or something like that, or on your homework. There is a couple of things that you should know before we begin for gene linkage. The first thing that you should know is that you should never use the branching method or matrix method to solve for a particular genotype or phenotype proportion from a cross, especially if all the genes are linked. So never, never, never use that, okay? So, and you know how many times I've seen students go back to using the branching method and or the matrix method to solve these problems. Never do that. Don't ever do that, okay? You will get it wrong. Because remember, the reason why we can't use those methods to solve these is because these genes are not independently assorting anymore. So we're not seeing every particular gamete proportion being equal. So therefore, we cannot use that methodology anymore, okay? So hopefully that made sense. Another thing that you should know is that for these problems, it is extremely critical for you to understand what the chromosomes are in the individuals that you're crossing together. So basically, what I mean by that is that if we take a look at the F1 over here, their genotype is big A, big B, little b, big C, little c. But the thing is, with that, you have to know what their chromosome alignment is. Basically, what each homolog has is very critical, and that'll change your final answer that you get for part A, part B, and for part C as well. It is extremely critical for you to understand that. But we're going to go over a bit more in detail like later, but it is extremely critical for you to understand what the actual genotype and what the chromosomes that you have for a particular individual. And that will sort of guide you towards the direction of how to solve these problems. Okay, so hopefully that made sense. And I think for now that should be everything that you should know. And one thing that you should also remember that because we have a linkage map given to us, we have it in map units. I recommend just writing down the recombination frequencies and the parental frequencies beforehand before you begin. So essentially what we have here is the recombination frequency between A and C is equal to 0.1. And then the recombination frequency between C and B is equal to 0.32. So the way how I'm getting recombination frequency is basically just the map units divided by 100. The map units can be written down as centimorgans as well. So that's basically how you're getting that. And to get the parental frequency, so parental frequency is just basically equal to 1 minus the RF, the recombination frequency. So the parental frequency between A and C is equal to 0.9. And then the parental frequency between C and B is equal to 0.68. So hopefully that made sense. And that's basically all we need for now, okay? So now what you want to determine first is figure out the chromosomes that you have, figure out each homolog, like if they have recessive alleles or if they have dominant alleles on one homologous chromosome compared to the other homologous chromosome, okay? The way how you might see this written down is as something like this. The professor might give you a genotype like this. So In this situation, what you have here is that this genotype of this individual has, on one of their homologous chromosomes, is basically big A, little b, and big C, and then on the other homologous chromosome, it's little a, big B, and then little c. And basically, you would use this map to figure out, you use this map in particular to figure out part A and part B and part C. On the other hand, what we have here, some professors might give you a more complicated problem where your parents originally were big A, big B, big C, or little a, little b, little c, and they're combining together to make the F1, correct? To make a all heterozygous individual in the offspring. So how can we determine that? Basically, we can say that this is the dad. The dad is giving a homologous chromosome that is basically just all big A, big B, or big C, big B. And then the mom, on the other hand, she's giving another chromosome that is all recessive. And you would essentially use this to figure out, you would use basically, there's two different types of ways that you might see these problems. You might see it as a slash, and the slash just basically means that this side of the genotype is basically one homologous chromosome, and then this is the other, and you would write it down as this. Or you might see another problem as we have it over here. Okay. Another thing that you should know is that you should always write down your linkage map that we have in the blue right now. You should always write it down as the orientation that you're given in the linkage map. Okay, so if your linkage map tells you that A is closest to C, then therefore your linkage map has to have A and C in order like that. Okay. You are not allowed to write it down as A, B, and C because in the linkage map, it clearly tells us that A is closer to C in terms of recombination frequency, okay? So hopefully that made sense so far. So what we want to do here is, this is going on into the next section of gene linkage, but let's say we had, as a quick little sidetrack, let's say if we're trying to cross big A, little a with big A, little a, if we're doing a Punnett square, for example, one of the things that a lot of students don't really understand is, if you haven't watched my videos before, but basically, you know, to get big A, it's basically a 1/4 probability, right? To get this big A, little a here is 1/4. 1/4 for big A, little a here, and then 1/4 for little a. Now the question is, how do we actually know that we're getting a 1/4 chance of big A, without just telling me that there's 4 squares there and there's only one possibility out of those 4 squares, right? So this is where a lot of students struggle on. Basically, from parent number 1, which is over here, to get the probability of getting big A gamete is essentially 1/2, right? Mom can either give the big A or a little a, and it's half chance, right? Same thing over here for dad. He can give a half chance as well. So what we will do is, normally we would multiply those together to get a 1/4 chance for big A, right? Okay. So what we're trying to do over here, on the other hand, we're trying to figure out a gamete proportion, which is big A, little b, and little c, or big C. We're trying to do the exact same thing, but except we don't know what our gamete proportions are for the F1, okay? So yeah, let's start working on that. Okay. So what we want to do is we want to figure out what is the probability of you getting big A, little b, and big C gamete, okay? So the way how you're going to do this, and this is extremely important why I told you to write down your linkage map, because what you want to do here is essentially you want to sort of keep track of what you want. You want a big A, you want a little b, and you want a big C. And basically you're going to connect those dots together. And essentially whenever you do a crossover event like between C and B as we have it here, therefore we're going to use the recombination frequency. But then when you stay on the same homologous chromosome like between A and C, we're going to use the parental frequency. Okay? So the equation that we'll do is to figure out what is the probability of getting this gamete. We would do, we're going to use the parental frequency between A and C, so 0.9. We're going to multiply that by the recombination frequency between C and B, which is 0.32. And then we are missing something though. One thing that we're missing is that when this recombination sequence happens that we have written out this way, what ends up happening is that we have one chromosome, the one that we want is big A, big C, and little b, correct? But at the same time, this sequence of events gives us another chromosome that also has little a, little c, and big B as well. So because of this, because out of this crossover event, we're getting two different products. We need to take into that account. So basically what we're going to do is you're going to divide it by 2. And that's basically it. That's how you solve for that. That's how you solve for that probability for that gamete. So hopefully that made sense. Okay? Okay, next one. So what we're going to do now is we're going to use the exact same linkage map that we have over here in the blue that we've developed. Let's write it down. But we have another issue in this problem though. Now in this problem, it tells you that you have an individual that has big A, big B, little b, big C, big C, little c. And you're trying to figure out the gamete proportion of big A, big B, and little c. But keep in mind, because we have two big A's here, what we're going to do is we're going to write down them. Both of their homologous chromosomes have to have a big A, right? And then the rest will be little c and then little b, okay? So that's the main difference between this problem and the last one. And here's another thing that you can also infer it. Because you have two big A's, the probability of you getting a big A, regardless of if genes are linked or not, it's still 100% to get big A. So in reality, the way how you can go about this problem is you can this big A probability is just 100% chance to get big A. So regardless of if you cross over or if you don't cross over, you're still getting a 100% chance of getting big A. So therefore A has nothing to do with this problem. Okay? Because we would always get big A 100% chance. So what you can do instead is this can be removed from the equation, and now you end up with this two of these C and B that you have to take into account, okay? So to figure out the probability of you getting big A, big B, and little c, to get big A, you have a 100% chance to get big A, right? To get big B and then little c, we're going to do a crossover event between those two. So we're going to use the recombination frequency between C and B, which is, I forget, 0.32. And then we're going to always divide it by 2, and then we will get our answer. And that is it. That is how you solve that problem, okay? So hopefully that made sense. That's sort of like a trick question that you might see on your exam. And then we have the last one, where we're crossing two heterozygous individuals, and we're trying to figure out how many offspring will have a certain genotype. Now, remember what I said, that you cannot use the branching method for this problem. You must use Punnett squares for this problem to figure out a certain genotype or phenotype proportion. Now, because we're using a tri-hybrid cross, there's a total of 64 squares that you have to fill out in order for you to do this problem. Now, most of the time, genetics professors are going to be nice to you, and they're not going to have you suffer through making 64 different possibilities to making a huge tri-hybrid cross. So they're going to give you a quick shortcuts that you can use to for these problems. So the shortcuts here are basically an individual that has 2 big A's and 2 little b's. Because they both have 2 big A's and 2 little b's, basically both parents have to give a big A and a little b. There is no other possibility that a parent can have for you to make that genotype. So therefore you can skip some of the gamete possibilities as well. So basically what I mean by that is that parent number 1, he has to give big A and little b, but parent number 1 has to give little c or little c, or big C and little c. So this is basically what I mean. There are no other possibilities for parent number 1 to other than these gametes to be made into that genotype. Okay. So same thing for the other one, the other parent. And then now what you want to do is you want to look for which of these squares satisfies our genotype. This one over here has 2 big C's. We don't want that one. And then this one over here has 2 little c's. We don't want that one. But these are the ones that we want, that we're going to take that into account. Okay. So same thing like what I mentioned before. We have a Punnett square. The way how we figure out what the proportion of a certain genotype is you need to figure out what the probability of their gametes are first, before we can multiply them together to figure out a genotype proportion. Okay. So what we need to do is basically we need to figure out the probability of that gamete and then this gamete as well. Okay. So let's do that. And keep in mind this individual is heterozygous. So let's do that. We're going to draw our linkage map again. You always need to draw your linkage map for these problems. Okay, so what we want is, for this one, we want a big A, we want a little b, and a little c. Now let me infer to the key that we have above. We're crossing over between A and C, so we're going to use the recombination frequency, so 0.01, or 0.1, excuse me. And then we're going to multiply that by the parental frequency between C and B, which is 0.68. And we're always going to divide these by 2, and then we're going to get that answer. So cool. Let me multiply that real quick. So we get an answer that is equal to 0.34 for the probability of you getting big A, little b, little c gamete. And then the other one that we want is probability of you getting big A, little b, big C gamete. So that one, on the other hand, we're going to do the same thing. Same thing. We want big A, we want little b, and we want big C. So this one is recombination frequency between C and B and parental frequency between A and C. A and C parental is 0.9. And then C and B recombination frequency, 0.32. And we're going to divide it by 2 to get our answer, which is I made a mistake over here. This one should be, oops, my bad. I made a mistake on my calculator. Yes, let me calculate this real quick. So this is 1.144. Okay, so now what we need to do is we have basically in this example, we have the gamete frequencies now. In order for us to figure out this 1/4 probability, you basically just multiply the 1/2 by the 1/2. And that's what we're going to do over here. So basically we're going to do 0.144 times 0.034. And this is the probability of you getting big A, little b, big C, gamete, or offspring. So that is equal to 0.144, 0.034. So you're getting something like 0.005. Yeah? Cool. So you're getting something like that, right? Cool. And we still have one more thing to focus on, is that remember that in this example that we have over here of our original Punnett square, we have two possibilities that satisfy the exact same genotype, right? We have two possibilities. We have a big A, little a from the right side, and we have a big A, little a from the left side, right? So two possibilities. So whenever you figure out a genotype like this, and there's multiple different possibilities that you can have to get the exact same genotype or phenotype, you basically have to use a sum rule in this case, okay? So because there's two different possibilities, one right here and one right here, and because we're essentially combining the exact same gametes again, we can essentially just multiply this by 2 and we get our answer. So 0.01. And that's it. That's how you solve these problems. Hopefully this helped you out. Yeah, and if you have any questions, just let me know in the comments section. And hopefully this helps you out. Bye.